Hello, welcome back to BB Lectures. This afternoon too, we are going to have a look at a very wonderful and interesting presentation. Body fluid evidence. That is the topic for this presentation. Body fluid evidence. So don't forget to help me grow my channel. At least appreciate what I'm doing, appreciate my efforts by subscribing, liking, sharing, and commenting on this video. And I'll also do very well to reply all your comments. Thank you. So, why are we going to discuss this topic? Or what's our objective? Our objective is to know how suspected body fluids are identified, collected, packaged, and analyzed in forensic investigations. With our introduction, we say body fluids are among the numerous biological evidence used in investigations. Every forensic process starts from the crime scene. Identification and analysis of body fluids is what we call forensic serology. That is one branch of forensic biology. And this serology that we are talking about precedes DNA analysis in a forensic lab. That is, when the evidence from the crime scene is body fluid, we first do serological testing before DNA analysis. So let's look at a brief history of serology or analysis of body fluids. So around 1900 or 1900, Karl Steiner discovered that there are 4 different types of human blood based on either the presence or absence of specific antigens found on the surface of the red blood cells. And this discovery led to the blood groups A, B, AB, and O. So in 1940, this same Steiner and Wiener reported the discovery of the Rh factor by studying the blood of the rhesus monkey. So what are body fluids? We are talking about body fluids. So one has to know what body fluids are. If someone asks you what are body fluids, you should be able to explain to the person what they are. So body fluids are liquids that originate from inside the body of humans. So anything liquid that comes from our body as human beings or Homo sapiens can be termed as body fluids. Example: blood, semen, saliva, urine, etc. You can add them. So among these body fluids that we are talking about, we've seen that blood is the most common. So please don't forget to subscribe to my channel. Appreciate my efforts. I spend a lot of time researching. Though I'm an expert in this field, I still research a lot. I put in much effort to bring these presentations. So appreciate my effort by subscribing, liking, sharing, and commenting on my videos or presentations. So let's take 20 seconds for each and everyone to subscribe. Let's go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, and 20. I hope you've all subscribed to my channel. God bless you and thank you very much. So let's continue with the presentation. But before you handle body fluid, let's say you find yourself at a scene of crime. You've gone through the whole process. You're about handling evidence. You are going to collect body fluid. There are some precautions or there are some measures that you have to undergo before you come into contact with these body fluids. 1. Consider all body fluids as biohazard. Consider each and every body fluid as a potential biohazard. So, by considering every body fluid as potential biohazard, you wear appropriate clothing. You don't enter a crime scene using your hands to touch body fluids. No. You wear appropriate clothing, coats, gloves, face mask, and then you change gloves before collecting each evidence. Why do you have to do so? To avoid cross-contamination. That's mixing the evidence, sending one evidence, especially the initial one you took, to the next evidence. So to avoid that and to mess up the work of the forensic DNA analyst at the lab, you have to change gloves before you pick up or before you collect each and every single biological or body fluid evidence. So anything biological, you change gloves before taking them. And since body fluids are also biological evidence, you do same. So, questions to answer when body fluids are encountered. When we pick up or collect body fluids, we ask ourselves 3 questions. Either you, the crime scene investigator, or the laboratory analyst. There are 3 questions that you have to answer. 1. The fluid that you've come across, is the fluid or stain blood, semen, saliva, urine, etc.? You might see something with your naked eye as blood because they look like blood. But you have to ask yourself, is it really blood? Is the fluid semen? Is it saliva? And this can be answered by what we call presumptive or the initial test. So after answering this question, you go on to answer the second question, which is also being answered with a second test. That is it of animal or human source? That is the body fluid that you've obtained. Is it coming from an animal or a human being? Then, once you're able to answer that with what we call a confirmatory test, you go on to your third and final question. That one can also be answered by a specific test. In modern times, it is answered by DNA profiling. And your third question is, can it be linked to a particular species or individual? Let's say you found blood. You tested it is blood. You went on to test. It was human blood. Now, can it be linked to a particular person? Can you say that particular blood is coming from person A or B? Or let's say it wasn't human blood. Can you link it to another species? Let's say a dog, an antelope, a cat, etc. Now, how to identify body fluids? How do we identify these body fluids that we've been talking about? That is at the scene of crime and also once the evidence gets to the laboratory. It depends on the jurisdiction. There are some countries when you go there, they identify these body fluids at the crime scene. And there are other countries, the crime scene investigators will just pick the evidence, send it to the lab. So the laboratory scientists now answer those three questions. So the first one is visual examination using your eye. So this is normally based on the experience of the analyst. This isn't admissible in a court of law. It can only give a clue to the analyst using your eye through your experience to know that, yes, this particular thing I'm seeing is indeed blood or not. It isn't admissible in a court of law because it is not scientific. The human eye can deceive, but it is still a way of identifying body fluids. Because you first even have to see it with your naked eye before any other form of test will come in. 2. Alternate light source. And this is of particular interest when you have hidden stains. Let's say blood in a dark garment. It is not easy to see it with your naked eye, so you use alternate light source to visualize. So normally when you use the alternate light source, the particular part of, let's say, a clothing or any other material that contains the stain will glow so that you can use that place for your test. 3. Presumptive test. This test tells the presence or absence of a suspected fluid, but it cannot confirm whether the fluid in question is from an animal or a human source. Example, blood. So when you get blood, you first do this presumptive test, but it's going to tell you whether it is blood or not. It cannot tell you whether it's animal blood or human blood. Then confirmatory test. This test gives a high degree of specificity for the body fluid in question. Example, human blood. That this kind of test can tell you whether the blood that you found is either human blood or animal blood, maybe a dog blood, cat's blood, monkey's blood, etc. So, alternate light source. This is used to visualize hidden stain, as I said earlier on, in clothing and other materials. So, after detection of stains, presumptive tests can then be performed. So a dark room is needed. So if you watch from the picture, they are in a dark room. They've protected their eye because these alternate light sources are not good for the human eye. Then where the stains are will glow for them, if it's a clothing, for them to cut that particular area for their test. So presumptive tests for blood. We are going to look at some presumptive tests for blood. Benzidine color test. So benzidine is carcinogenic, so not often used. So how is it done? You combine benzidine plus the stain in question, the blood stain, then you add hydrogen peroxide. If it turns to a pink color, then it's a positive result. Carcinemic test. You add phenolphthalein plus the blood stain plus hydrogen peroxide. If you get a pink color, then you have a positive result. Luminol. Luminol reacts with blood to produce luminescence, that it glows. So once the blood comes into contact with luminol, it glows or it emits light so that you can know that this particular place is where the blood is. is mostly used for hidden blood stains. Hematase test. These are reagent strips with tetramethylbenzidine ends. And TMB changes from orange to green when it comes into contact with blood. So once you apply this hematase strip to a blood stain, let's say you have wet blood in its liquid form, you just apply it to it and it turns from orange to green, then you can say you have blood. If you have a dried blood stain, it means you have to wet the strip. Then you apply it to your stain. Once again, if it changes color from orange to green, then you can boldly say that you have blood. So this is a hematase strip. This is how the strip looks like with an orange end. If you look at this picture, it means it has come into contact with blood, suspected blood. If you look at this picture too, luminol has been used. So originally, this is how the basin looks to the naked eye. Then once you put off the lights and you spray the place with luminol, you are getting this glow, the blue color. It means this place contains blood, but then maybe the perpetrator or whoever came into contact with the blood cleaned the blood. So over here, the blood was hidden, but with luminol, the place had glowed. This is the phenolphthalein test. You can as well see this is a negative result where you are not getting any color change on the swab, and this is a positive result. So I'm still reminding you to subscribe to this channel for me. Help me grow my channel. Appreciate my effort. Help me grow my channel by subscribing, liking my videos, sharing, and giving your comment in the comment section. So let's give ourselves 10 seconds for each and every one to subscribe. Let's go. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Well done to each and every one of you. I hope you've all subscribed to my channel. Thank you. And we can now continue with our presentation. So let's go on to presumptive test for semen, another body fluid. So we have the acid phosphatic test. And this acid phosphatase is an enzyme which is secreted by the prostate gland into seminal fluid of semen. Several chemicals detect AP in semen, and this alpha naphthyl phosphate, it turns purple when it comes into contact with semen. Fat blue B dye also turns purple when it comes into contact with semen. 4-methyl amblyferol phosphate, it glows when it comes into contact with semen. Now, research has proven that there is 400 times acid phosphatase, that's this enzyme, in semen than any other body fluid. So with this test, reaction time of less than 30 seconds means that you have found semen and not any other fluid or not any other thing. So this is the acid phosphatase, this is the container, and these are not strips. They are like some pieces of hard paper. So if you have a dry, let's say dry semen stain, you wet the ends of these ones. You open it. This is how it comes, but you open it like this. You wet it, then you apply it to the semen stain. If it changes color to purple, then you have semen. So that's a negative result, and that's a positive result over there in the picture. Test for saliva. This is called the amylase test. And amylase, as we all know, is an enzyme found at high levels in saliva. Now, this amylase test is currently the most widely used method for testing saliva. Amylase is also found in a variety of body fluids: saliva, blood, urine, sweat, tears, semen, breast milk, feces, and vaginal secretions, but is more concentrated in saliva. Fabulous and rapid stain identification of human saliva kits used by many crime labs for presumptive and confirmatory tests. So these are commercial kits, as you can see from the picture. So you apply saliva to the cassette over here. It has a control and a test line. Once what you are testing is saliva, the control and the test lines will appear. If it is negative, only the control line will appear. Test for urine, presumptive test. This is called the Jaffe test. Creatinine can serve as a screening test for urine. So how do you do this? To a drop of stain extract on filter paper, you add 1 drop of picric acid, followed by 1 drop of 5% sodium hydroxide. And if you get a brown or orange color, then it means the presence of creatinine. Then you can also say you have urine. So we come to the end of presumptive test. We are moving on to confirmatory test. After presumptive test, you move on to confirm your body fluid. So let's look at confirmatory test for blood. Precipitin test. How is it done? Combining human antiserum and human blood gives agglutination or precipitation. And this principle has been employed in the production of commercial kits. Example is the hexagon OBTI kit. This test requires only small amount of blood, and you can work with dry blood stain as old as 10 to 15 years. So this is the hexagon OBTI kit. This kit was used to test the sample here. That is a shorts clothing material soaked with blood. Now before the test, the analyst didn't know whether it was human blood or animal blood. So to make sure the kit was working, analyst performed a positive control. That's applying a known blood to the kit, and he got a positive result. He also did a negative control using the reagent blank, also as a form or to show that the kit was working to perfection. He now came to apply the sample, the extract of the blood, to the kit. As you can see, the test line showed over here, the controlled line also showed, meaning a positive result. Then electrophoretic method. How is it done? So human antibodies and blood stain are placed in wells on electrophoretic plate. Now electric current is used to move antibodies and antigens towards each other. And we know once the antibodies and antigens move towards each other and there is agglutination or precipitation, then you can say it is human blood. Now let's move on to confirmatory test for semen. Prostate specific antigen test, that's the PSA test. PSA is a protein produced by cells of the prostate gland and is produced for the ejaculate, that is what comes out of the male, where it liquefies semen and allows sperm to swim freely. Now PSA is also present in small quantities in the serum of men with healthy prostates. So this is a PSA kit or cassette. And this picture over here, the kit was used to test for suspected human semen in pajamas. So this is a positive test. So the analyst used two kits. One is a strip, the other one is a cassette. And there's another cassette here too, another one down here, all showing positive results. Now, collection of body fluids at crime scenes. We've been talking about body fluids. How are they collected at the scene of crime? Because you have to collect them before you can go through the analysis that we've been talking about. You can either use dried swabs, that is, if the body fluid is in its natural liquid form. You can also use wet swabs if you have a dried body fluid. You can scrape if the fluid is maybe on a wall or a surface. You can cut if it's in a clothing or material that you can cut. Then you can collect the evidence as a whole. Maybe you have a bedspread. You can package everything if you do not want to cut the area that contains the body fluid. But before you collect every evidence at a crime scene, there is a general rule. You photograph every evidence before collection. This is a basic rule in crime scene processing. So after collecting these body fluids, how do we package them? So packaging of body fluids. Now all swabs, that is whether dry swab or wet swabs, after collection of body fluids must be packaged in paper envelopes. Our emphasis is on paper envelopes. Do not use plastics because the heat from the plastics plus the moisture will degrade your DNA in the biological evidence that you've collected. So always try to avoid plastics. Use paper. All liquid evidence must be put in appropriate container and frozen. If you have liquid evidence, you put them in an appropriate container, like maybe EDTA tubes or any appropriate container, and freeze them. Once this is done, you have to transport them as soon as possible to the forensic laboratory. If you don't want this to, you can take a swab of this liquid evidence, air dry them, and then package them once again in paper envelopes. If you want to go for the second option, then you can use ice packs. So you place the evidence on ice packs and transport them to the forensic laboratory. And package every evidence separately with appropriate label. Do not package two biological evidence together. You don't know whether they are coming from the same source or not. They might be coming from two different individuals. So under no circumstance should you package two or more biological evidence. In this case, we are talking about body fluids together when you happen to find yourself at a crime scene. And also labeling. This is also a general rule in every crime scene process. You label every evidence appropriately before you transport them. So conclusion. Gradually, we've come to the end of our lecture or presentation. We say identification and analysis of various body fluids is paramount in the investigation of many crimes. There are a lot of crimes that forensic evidence is key to unraveling its mystery. So the collection, packaging, and storage of body fluids is key to successful DNA analysis. If you don't collect them well, if you don't package them well, and if you don't store them well, no DNA analyst can cajole magic to get a good result. There's an adage that says, garbage in, garbage out. So if you find yourself at a scene of crime, you come into contact with body fluid evidence, and you don't collect, package, and store them well, then the forensic analyst at the lab will only work on the evidence, but the result he gets, there was nothing he can do about it. So everything starts from the crime scene. Collect your evidence well, package them well, and store them well. When you transport them to the forensic lab and the analyst is not ready to work on them, he or she will also store them at the appropriate temperatures for serology and DNA analysis. Folks, once again, do well to subscribe to my channel. Help me grow it. I put in a lot of effort in bringing these lecture presentations. So what you can also do to help me is to subscribe, like, share, and leave your comment. I know most of you are experts in this biological field, forensic science, biotechnology, biochemistry. So when you come, you can ask your questions, you can leave your suggestions, your comments. Anything is welcome. And I also try to follow it up. Thank you very much. So your questions can be left in our comment section. You leave every question you have in my comment section. So this is a lecture presentation from BB Lectures. Thank you. And do come back, follow up to our upcoming lecture or presentation. We are going to have an overview of a forensic science laboratory, the various sections, what they do at the various sessions, etc. To be an interesting presentation or lecture. So don't miss it. Come back to our channel. Invite others to come and subscribe so that they will get every video. There are upcoming lectures and presentations that will be of interest to each and everyone, whether scientists or non-scientists. Thank you very much. I'm looking forward to welcoming you back to my channel. Once again, thank you.